In this video, we're looking at a brief overview of the SCGE Toolkit. The Somatic Cell Genome Editing Consortium, or SCGE, is an NIH Common Fund program that aims to develop safe and effective methods to perform gene editing to treat genetic diseases in somatic cells. In Phase 1, the SCGE worked to improve the efficacy and specificity of genome editing approaches. The program has developed a genome editing toolkit to disseminate novel strategies, technologies, and learnings to the broader scientific community. The toolkit contains a lot of information from consortium researchers, and there are different ways to view this information. The most broad way to view information is looking at projects. Projects are the overarching projects that PIs worked on throughout Phase 1 of the SCGE. Within the projects, researchers completed various experiments. You can choose to view individual experiments instead of the entire project. Within experiments, researchers studied and tested various objects. This includes genome editors, model systems, delivery systems, guides, vectors, and protocols. Each of these highlighted terms is a different way to view information in the toolkit. Now, let's take a look at the actual toolkit. This is the homepage. At the top, there's a search bar to search for different terms or investigators. Below that is the main menu bar, which links to each of the categories previously mentioned. Then there's a short blurb explaining the toolkit, as well as buttons to learn more about the toolkit or more about the SCGE program. And if we scroll down, this explains more of that toolkit organization that we previously covered. So now let's do a quick example of how you might search for information in the toolkit. Let's say you're interested in delivery systems. So we're going to go back up to the main menu and click on that category, and this is going to bring you to the delivery systems page. As you can see, here are the results, and you can view them in table view or list view. Now we're going to go back to table view. On the left, there are filters that allow you to filter the results to narrow down your search by a variety of topics. So let's say that you're interested in nanoparticle. So we'll click on that box, and that's going to automatically filter your results. You can see there's much fewer results in this time. And then we're going to click on the first link because we'll say you're interested in this specific delivery system. So at the top of this page, there's information about the delivery system that you clicked on. And if you scroll down, you can see which project and which experiments that this specific delivery system was used in. And all of these links are clickable. So let's click on this first experiment here. This is going to take you to the experiment page. At the top, there's a little bit more information about the experiment, and if you scroll down, you can see which organ systems the experiment is relevant for. And then let's just say, since this is only in the one system, we'll click that, and now we're going to click View Selected Tissues. And that's going to pull up some results and data sets from this experiment that you can learn more about. But now let's say that great, you saw this experiment, but you would like to learn more about the overarching project that this experiment is part of. So we go back to the top, it says parent project here, and it links to the original project. So we're going to go click on that. This is the project page. And as you can see at the top, it talks more about the project broadly, as well as links to the NIH reporter page. And if you scroll down, you can see the different experiments that are within this project. So it really ties together nicely. You can go very broad, look at the projects, and then you can go really in depth and look at those specific genome editors, model systems, and different objects that you want to learn more about. There's a lot of great information for you to explore. You can view the toolkit at scge.mcw.edu/toolkit.